Hello and welcome to M.D.Zafar lectures, where I try my best to make medicine fun to learn. If you are new to my YouTube channel, kindly subscribe and share my channel with your friends as well. So today we are going to talk about saliva, and saliva is produced from the salivary gland. So first of all, we will talk about their division as well as their structure. Salivary glands are of two types, the major salivary glands and the minor salivary glands. The major salivary glands include parotid, submandibular, and sublingual, whereas the minor salivary glands, which are abundant in quantity, are labial and buccal gland, palatine, and lingual glands. These are just the special ones. Now, further talking about the major salivary glands, all of them are paired salivary glands. Now we will have an overview about the histology of these major salivary glands. Parotid is a serous type of salivary gland, whereas submandibular is mixed serous as well as mucus, and sublingual type of glands are mucus. Now we will discuss what type of production makes them serous or mucus. Now a gland is labeled as serous if it has an enzyme, and the salivary gland, that is parotid and submandibular, both produce alpha amylase in certain quantities which acts on starch and breaks it down into maltose and glucose. Whereas a gland is predominant with mucin is labeled as mucus producing gland, and the major function of mucin is to act as a lubricant and help to soften the food and also to help in the process of swallowing. So now we will talk about the structure of salivary gland. Each of the salivary gland has three main components: the acinus, the myoepithelial cells, and the branching duct system. I will explain these components of salivary gland through this diagram. Now, as you could see, on one end there is a grape-like structure which is a blind end, and then we have a duct system. This grape-like blind end is known as acini. Now, acini has two types of cells. On the inside, we have the acinous cells, and on the outside, which are orange cells, these are the myoepithelial cells. The primary role of the acinar cells present on the inside is to produce saliva, and this initial salivary production is isotonic with the plasma. Once the saliva has been produced, the myoepithelial cells present on the outside will contract and pushes the saliva from the acini into the duct system. Now, this duct system is lined with the columnar cells and it is impermeable to water. As saliva will move through this duct system, different ions will either be reabsorbed or secreted. Now, the most important mentions among the secreted ions is potassium and bicarbonate, whereas the ones which are reabsorbed are sodium and chloride. So as a result of this change in the composition, saliva eventually becomes hypertonic. Saliva production has two types of flow rates, a high flow rate and a low flow rate. At high flow rate, that is more production of saliva, it will pass through more rapidly through the duct system as a result, less absorption and less secretion will take place and saliva will remain isotonic to plasma. That is, it will have high concentration of sodium and chloride and low concentration of potassium. But at a slow or low flow rate, when there is less production of saliva, more sodium and chloride will be absorbed, whereas potassium and bicarbonate will be secreted, and as a result, the saliva will become hypotonic. So now we will talk about the regulation of saliva production. Saliva production has a unique control because both parasympathetic nervous system and sympathetic nervous system acts to increase the production of saliva, but their mechanisms are different. However, there are other factors which influence or inhibit both these types of the autonomic nervous system in the production of saliva. First of all, we will talk about the factors which influence the production of saliva, and they include presence of food, smell, nausea, or conditioned reflexes. On the presence of any of these factors, the cranial nerve 7 and 9, namely facial and glossopharyngeal, part of the parasympathetic nervous system will carry the information to the salivary glands, and at the end point, acetylcholine will be released from the nerve terminals. It will act on the muscarinic 3 receptors and within the acinar and ductal cells through IP3 and calcium being the second messenger, saliva will be produced. These factors will also influence the sympathetic part of the nervous system, and it will produce saliva by the production of norepinephrine, which will act on the beta receptors present on the acinar endocrine cells, whereas the second messenger in the case of sympathetic nervous system is cyclic AMP. Now, fear, sleep, and anticholinergic drugs like atropine will inhibit both parasympathetic as well as sympathetic nervous system and will eventually decrease the production of saliva. So, as we have learned about the production and regulation of saliva, now we will talk about its composition and its functions. Saliva is composed of alpha amylase and lingual lipase, and their primary function is to initiate the starch and triglyceride digestion within the mouth. Saliva also has 90% water and mucin, and the main function of these two components is to provide the lubrication. Then saliva also has IgA antibody and lysozymes and lactoferrin. The function of these three components is to provide protection. IgA antibody is a part of innate immunity. Lysozymes disrupts the bacterial cell wall, and lactoferrin removes iron from bacteria and prevents their growth within the mouth. So, thank you very much for listening.